Okay, the third and last application I want to discuss is the application of magnetic nanoparticles in biomedicine. Magnetic nanoparticles can be manipulated by external magnetic fields. That means site selection is possible. We can put them where we want them to be. They have widely tunable sizes from and the diameter of a magnetic nanoparticle is less than that of a cell. 100 to 200 nanometers. Viruses have dimensions of 20 to 450 nanometers. Proteins have dimensions 5 to 50. And a gene is about 2 nanometers wide and 10 to 100 nanometers long. So you can interface magnetic nanoparticles with a whole variety of biological entities. These can be coated with biological and chemical molecules, which means you can do tagging and drug delivery. They can absorb magnetic field energy, which means you can use them for contrast enhancement and magnetic resonance imaging. This is already being commercially done, and so I will not go into this at all, because this is now not an area of research. This is common practice. The current areas of research are magnetic hypothermia, through which one can enhance radio and chemotherapies, and heat-assisted drug delivery. Okay, so to be able to use magnetic nanoparticles in biomedicine, one needs to put them inside the body. So one needs to understand what happens to magnetic nanoparticles once they are inside the body. There are several issues. The first one is getting magnetic nanoparticles to the desired site. So direct injection into the bloodstream or site of treatment is one option. And of course you can use appropriate magnetic fields and field gradients for targeting, or to get the magnetic nanoparticles where we want them to be. The second issue is ensuring the survival of magnetic nanoparticles in the body for the required time, because you know that your body will reject any foreign object. That is part of the body's defense mechanism. So we need to make sure that these magnetic nanoparticles become invisible to our defense mechanism for the time that they're required to conduct their job. So suppose this is an artery and we have injected a magnetic nanoparticle here by intravenous injection. The first thing that happens is a process known as opsonization, which is that magnetic nanoparticles get surrounded by certain plasma proteins, which makes them detectable to the body's defense mechanism, which is made up of phagocytes. So phagocytes are cells which will come and encapsulate this magnetic nanoparticle and make sure that it is chucked out from the body. So what one needs to do to use magnetic nanoparticles for biomedical application is to inhibit the process of opsonization, because that is what makes the nanoparticle visible to the phagocytes. So what one does there is you coat magnetic nanoparticles with hydrophilic polymer chains, which will prevent the opsonization process, such as dextran, polyethylene glycol, polyacrylic acid, etc. And this inhibits the opsonization and ensures larger circulation times. The third issue is that the magnetic nanoparticle, or the material we're using, should have no or minimal cytotoxicity for healthy cells. One should be able to remove it safely from the body, and it should not deposit in organs like the spleen or liver. And it is known that gold and silver nanoparticles and metallic nanoparticles have a tendency to doing that. And actually the only material which is officially approved by the US Food and Drug Authority, the FDA, is magnetite or iron oxide for use inside the human body. And all other materials are now being researched, and other ways are people are trying to find ways to make them biocompatible. So what are the general requirements for magnetic nanoparticles? It is desirable to have superparamagnetic nanoparticles, first because they have a very small size, second because after we've gotten them to the place where we want them to be, we've done whatever we want them to do, then you want to be able to remove them. And superparamagnetic nanoparticles have zero remnants, so that when you remove the magnetic field, which was applied either for their targeting or for any other job, then they lose the magnetization, so they do not stick to one another, they will not agglomerate. They have a diameter of 5 to 10 nanometers, which is suitable for most applications. They have a large, what is required is a large magnetic susceptibility at body temperatures. Susceptibility is the ratio of magnetization and magnetic field. So a large susceptibility simply means that in a given magnetic field, a large magnetization is to be produced. And of course, last but not least, they should be biocompatible. The applications that we'll be looking at are magnetic drug delivery and magnetic hypothermia. Currently, an application that is currently in standard practice is contrast enhancement in magnetic resonance imaging, but we'll not go into that because that will require understanding a lot of physics, and it is already standard practice. Okay, so magnetic targeting therapy. Let's look at drug delivery. The motivation is to minimize harmful effects of cytotoxic therapies on healthy cells by delivering the cytotoxic drug to the required site. So instead of putting a drug as is done in chemotherapy, putting a cytotoxic drug is one that is harmful for healthy cells. So instead of exposing the whole body to it, as is currently done in chemotherapy, what one does is that one delivers that drug specifically to the site where it is required, which may be the site of a malignant tumor. So this leads to a localized administration of the drug, and it also leads to reduced dosage. So what you have here is a magnetic nanoparticle in black. It is then coated by suitable polymers, functionalized, so that we can attach these blue drug molecules to it. This is then injected into the artery by intravenous injection. This is an artery which is supplying, or a blood vessel which is supplying the area where we want to go, which may be a tumor, for example here. It is directed to the site using magnetic fields, and then you can use different means, for example, a change in temperature via infrared heating of the body, or a change in pH through some other locally administered chemical, to break these functionalities, to break these bonds and release the drug. And this drug can then be administered directly to where it is required. The alternative approach is a magnetically controlled drug implant. This was also patented very recently, just about a year or two ago. Here you have a drug reservoir. So this is a chip which is put inside the body near to the area where the drug is required. There's a drug reservoir, and this is the yellow is a porous membrane with a nanogel. So the black circles here are a nanogel, which is inserted into the pores of this membrane, and the dark blue circles are magnetic nanoparticles. When we expose this to a magnetic field, a radio frequency magnetic field, the magnetic nanoparticles heat up through various heating mechanisms, which we'll be discussing, as a result of which this gel shrinks and the drug is released. And this process is reversible. When we release the heat, the gel expands again to its original size, and the drug reservoir is closed. So that is an alternative heat-assisted drug delivery therapy. The last application I want to discuss is magnetic hypothermia. This refers to heat generated by magnetic nanoparticles in an AC magnetic field, and it has applications in therapeutics, in particular in cancer therapy. The ideal temperature for hypothermia treatment is 42 to 45 degrees centigrade, which is equivalent to 315 to 318 Kelvin. Temperatures in excess of 45 degrees centigrade can cause harmful side effects, for example, damage to healthy tissue. And it has been found that at these temperatures, 42 to 45 degrees centigrade, the DNA of malignant cells is destroyed, while the DNA of healthy cells remains intact. So if we can heat the malignant cells up to these temperatures, then we can effectively kill cancer cells. So at these temperatures, you can have direct necrosis of cancer cells, and it can also produce an enhanced temperature can also produce increased sensitivity to chemo and radiotherapies. So in order to understand hypothermia and the modalities of hypothermia, we need to look at heating mechanisms in magnetic materials. Heat is generated in an AC magnetic field due to different loss mechanisms. One is hysteresis losses, inductive losses, relaxational losses, which can be subdivided into Brownian and Near losses, and ferromagnetic resonance losses. I've put the last in brackets because these occur at gigahertz frequency ranges, which are not suitable for biomedical applications because they can cause neuromuscular responses. Important considerations for hypothermia treatment are that the nanoparticles or the magnetic medium that is being used for hypothermia or as an agent of hypothermia should have a high specific loss power. So the magnetic nanoparticles should have this ability to generate a large amount of heat per unit mass so that you can minimize the dosage. So the specific loss power is basically the amount of heat generated per unit mass of the magnetic nanomaterial. There should be minimal damage to healthy cells by heat or by the AC field. There should be a precise control over the heating rate and duration. Hysteresis losses occur in ferromagnetic particles, which are about 500 nanometers to 1 micron in size. And the change in internal energy per cycle of magnetic field is given by the integral of MNH. And the amount of energy generated is given by the area of the magnetization hysteresis loop. If I have an AC magnetic field, then I can write it as H naught, which is the amplitude cosine omega t, where omega is the frequency of the field. And in this case, the magnetization can be written as H naught chi prime cosine omega t plus chi double prime sine omega t. Here chi prime and chi double prime are the in-phase and out-of-phase parts of the magnetic susceptibility. So substituting both H and M in the first expression, we get that the amount of energy generated per cycle can be written as this integral, and the power dissipated per cycle is mu naught pi chi double prime, which is a function of frequency, times the frequency and the amplitude of the applied field squared. So here f h naught squared is the external parameter of hypothermia, that is the frequency and amplitude of the RF field that you're applying. The magnetic properties of the material are contained here in the out-of-phase part of the susceptibility. So high temperatures are achieved at frequencies in the kilohertz to megahertz range, but for hysteresis losses you need large particle sizes, which produces the problem of agglomeration, and there's only extrinsic control of heating is possible, which can cause damage to healthy tissue. Inductive losses occur in high resistivity metals due to eddy current heating. And for biomedical applications, previously, about maybe 20 years or so ago, thermoseeds were used. So these were metallic objects about a centimeter in dimension, which were surgically implanted in the area to be treated. They were then exposed to an AC magnetic field, and they produced eddy current heating, and then they had to be surgically removed. However, treatment by inductive losses have several disadvantages, namely that the thermoseeds are large and they need to be surgically implanted and removed, and again only extrinsic control of heating is possible. On the other hand, there are relaxational losses where we have Brownian and Near losses. The magnetic relaxation is defined as the change with time of magnetization from its initial value at T naught divided by a certain characteristic relaxation time. The Brownian relaxation occurs due to rotation of a ferromagnetic particle in a viscous fluid. So if I have a ferromagnetic particle, it is magnetized. If I place it in an AC magnetic field, then it will rotate, and its physical rotation will generate friction in a viscous fluid, and this generates the heat which can be used for hypothermia. The Brownian relaxation time is given by this expression here, where eta is the viscosity and V is the hydrodynamic volume of the particle. The losses that are most important for in current day hypothermia research are nail relaxation losses, and they occur due to superparamagnetism in particles which are less than 10 nanometers in size. So here, when you put a superparamagnetic nanoparticle in an AC field, the nanoparticle itself does not rotate, but its magnetization flips over the anisotropy energy barrier, and because the magnetization is coupled to the lattice, this change in direction of the magnetization generates phonons, and so heat is generated. So the nail relaxation time is proportional to the exponential function of anisotropy constant times volume divided by the available thermal energy. And since both nail and Brown relaxations occur in parallel, there is an effective relaxation time given by the sum of the reciprocals of both relaxation processes. And the power dissipated due to relaxation processes, assuming monodispersed particles with a constant value of the susceptibility, is given by this expression here. So you can see that the magnetic properties are included first in the susceptibility here, and secondly in this relaxation time here, because here you have the anisotropy constant and the volume. So the physical properties of the particle go in the value of tau and in susceptibility. This is a graph which shows how the relaxation times vary with particle diameters, and the Brownian relaxation time is a very slowly varying function of particle diameter, and while the nail relaxation time varies quite rapidly with particle diameter. So at large particle sizes we can see that Brownian relaxation dominates, and at small particle sizes or at intermediate particle sizes we will have nail relaxation dominating. So in all these processes there is the possibility of overheating or spot heating due to localized heating, and this can cause damage to normal tissue. However, if somehow we can control the Curie temperature to lie in the range which is known as the therapeutic limit, which is 42 to 46 degrees centigrade, then the particles lose the magnetism at Tc and stop heating. So remember, I told you in one of the first slides that the Curie temperature is the temperature at which the transition from a ferromagnet to a paramagnet occurs. So this is the temperature where the exchange coupling which defines the ferromagnet breaks down, and you have decoupled moments which do not generate any significant amount of heat. So if we can engineer this Curie temperature to lie in this range, then the particles can act as automatic heat switches, which is with a switching temperature which is equal to the therapeutic temperature. And in this manner harmful side effects can be minimized. This is the modality known as self-controlled hypothermia. So what is needed for that? We need ferro or ferrimagnetic nanoparticles with large magnetization, Curie temperature lying in the range 42 to 46 degrees centigrade, and a minimal cytotoxic effects on healthy cells. And this is quite challenging because both the magnetization and the Curie temperature vary in the same manner. However, what we require here is large magnetization and low Curie temperature. It is generally seen that the magnetization and the Curie temperature they behave in such a manner that materials with large magnetizations have large Curie temperatures and vice versa, because they both depend upon the strength of the exchange interactions. So in order, what is required now is that they move in opposite directions, and for that one needs to employ several techniques: particle size manipulation, coating the particles with other materials, and so on and so forth. So I'll just give you an example of some hypothermia measurements which we have made on lanthanum, strontium, manganite nanoparticles in our lab at a frequency of 214 kilohertz and a maximum field of 24 oersteds. And I must mention here that in order to have materials with high magnetization and low Curie temperatures, one route which is being very actively investigated is to use tunable magnetic systems. Tunable magnetic systems are those in which you have magnetic interactions which are mediated through non-magnetic material ions, for example oxygen ions, as is the case in magnetic ferrites or in manganites. And in these materials, by tuning the magnetic interaction through doping with various materials, you can control the strength of the interaction, and so you can control the magnetic properties. So what is required really now is a tunable magnetic system, and lanthanum strontium manganite is one of those magnetic systems where you can change the value of X, and the Curie temperature of this system lies in the range required for self-controlled hypothermia. So here you can see, as you change X from 0.15 to 0.45, the heating varies non-monotonically. It increases, and then decreases, and then increases. And these are all samples with the same average particle size, so we are basically only tuning the magnetic interactions here. And the specific absorption rate, which is the amount of heat absorbed per unit mass of nanoparticles, is given by the heat capacity of the nanoparticles times this derivative here, which is the slope of the heating curve at time t equal to 0, and the ratio of the sample mass to the mass of the magnetic component in the sample, which in this case is the manganese ion. And here we have the specific absorption rates, and we can see that in this tunable magnetic system, the specific absorption rate varies non-monotonically with the strontium concentration. And you can achieve temperatures ranging from what is required for self-controlled hypothermia, which is about, let's say, 46, 47 degrees centigrade, to almost 65 degrees centigrade, which is the modality of hypothermia known as thermoablation.